Hello students, welcome to EPG Pathshala. I am Dr. P.M. Reddy from the Energy and Resources Institute. Today we are going to talk on module cloning strategies under the paper Plant Biotechnology and Crop Improvement. Learning objectives of this topic include isolation of the DNA fragment containing the gene of interest, processing of DNA fragment, with restriction endonucleases and DNA modifying enzymes, and insertion of isolated DNA fragment into vector using ligation process, introduction of DNA into host cell, which is known as transformation, and the selection of transformed cells. What is actually gene cloning? Gene cloning is the basic strategy that is used to obtain a recombinant DNA molecule. A recombinant DNA molecule is a vector, for example, a plasmid or phage or a virus, into which the desired DNA fragment has been inserted to enable its cloning in a suitable host. Why we perform cloning of a gene? A particular gene can be isolated and its nucleotide sequence can be determined. Once you determine the nucleotide sequence, protein, enzyme, or RNA function can be ascertained. Mutations can be identified, for example, gene defects related to specific disease, and so on. Organisms can be bioengineered for a specific purpose, for example, insulin production, pest resistance, etc. There are four fundamental steps in gene cloning. They include isolation of desired gene, which is to be cloned, insertion of isolated gene in a suitable vector, introduction of this vector into host bacterium, which is known as transformation. In this, the vector multiplies within the host cell, producing numerous copies of recombinant DNA molecule, as shown here on the right side. Copies of recombinant DNA molecules are passed on to progeny during host cell division, and a colony of host cells carrying recombinant DNA is produced after a number of cell divisions. You can see here there are two cells at the bottom. They contain several recombinant DNA molecules in them, starting from a single one at the top. And the step 4 is, once the cells are transformed to the vector, how they can be selected. That is, selection of transformed cells.
Ready?
Here, the cloning strategies involve different steps. The first step is isolation of a DNA fragment containing the gene of interest to be cloned. Identification and isolation of desired gene or of DNA fragment is a crucial step in gene cloning. DNA fragments for cloning are obtained as follows. If you have the DNA, they can be restricted by using restriction endonuclease digestion of genomic DNA. After that, construction of cDNA library, and also through direct chemical synthesis, a DNA molecule can be synthesized, and the polymerase chain reaction. These are the different steps which can be used to isolate DNA fragments. What are restriction endonucleases? These are the enzymes that produce internal cuts in a DNA molecule. Restriction endonucleases are defined as indispensable for DNA cloning and sequencing. They serve as tools for cutting DNA molecule at predetermined sites, which is the basic requirement for gene cloning. Only type 2 restriction endonucleases are used for gene cloning in view of their cleavage only at a specific site. This particular slide shows different types of restriction enzymes and their recognition sites. Restriction enzyme names or examples are denoted in first column, and their recognition sequence are denoted in third column. If you can pay your attention to column 4, you have two different types of molecules which are generated have a sticky ends, and the molecules which are generated due to action of a particular resistance enzyme can be blunted. Two examples are given here at the bottom of the slide. For example, restriction enzyme ACOR1 it recognizes a particular sequence, that is GAATTC, and these restriction sites are palindromic. Both upper strand or lower strand or plus strand or minus strand, they contain same sequence. If they read 5' to 3'. ACOR1 makes a staggered cut as shown by arrowheads here, thereby cleaving the DNA molecule into two, with two DNA molecules having overhangs that are sticky ends as shown in this picture. Another example is shown here is restriction enzyme SMAW1, which recognizes a sequence which has CCC, GGG nucleotides, 5' to 3'. This makes a single cut in both strands between C and G, thereby creating blunt-ended molecules. Restriction endonucleases, as I already mentioned, they are very important in gene cloning. They are very powerful tools. There are certain other enzymes which are used in gene cloning. They are also extremely important. For example, these are also called DNA modifying enzymes, which include, first, alkaline phosphatase. This enzyme removes the phosphate group present at 5' terminus of the DNA molecule, as shown in the first row of the picture on the right side. You can see the phosphorus molecule at the 5' end, due to the action of alkaline phosphatase, are converted into OH sites. Phosphate is removed. Another modifying enzyme is polynucleotide kinase. What this enzyme does is, it has a reverse effect of alkaline phosphatase. This adds phosphate group into free 5' end. If a DNA molecule is not phosphorylated, that can be phosphorylated by this polynucleotide kinase. Thereby, adding of phosphorus facilitates gene cloning or forming phosphate bonds. Third, DNA modifying enzyme, which is used in terminal deoxynucleotide transferase. This adds one or more deoxyribonucleotides onto the 3' terminus of a DNA molecule, as shown in the figure here. Like one can generate at the 3' end, for example, poly T or a poly C, and so on. Normally, poly T is used so that it can anneal with poly A of a template transfer. What is a polymerase chain reaction, which is in short known as PCR? PCR is the most commonly used technique to purify a gene or a DNA fragment. This is developed by Kerry Mullis in 1985, for which the scientist got a Nobel Prize. It produces micrograms of DNA copies of the desired DNA in a matter of few hours. The amplified DNA fragment having a gene of interest is extracted, purified using suitable gel extraction kits. Polymerase chain reaction, it is carried out in vitro. The things or the items which are needed for PCR reaction are, we should have a sequence information, sequence of a DNA molecule, oligonucleotide primers. Primers are nothing but a small fragments of a DNA, the single stranded DNA, comprising of about 20 to 30 bases, and a template DNA onto which a polymerase chain reaction is performed to generate more copies of this same DNA, and need a heat-stable DNA polymerase, it is a Taq polymerase, and also we need DNTPs, nucleotides, deoxynucleotide phosphate, and suitable buffer. For PCR reaction, all these ingredients which were described earlier, they are mixed together along with the DNA template, and a reaction is performed. This PCR reaction consists of is performed in three different stages while it's going. First reaction is denaturation. Double stranded DNA is heated to 94 to 95 degrees to separate it into two single strands. The high temperature causes hydrogen bonds between the bases to break and the two strands separate. It usually takes about 15 to 30 seconds. The two separated single strands act as template for production of new DNA strands, as shown in the figure at the bottom. A double-strand DNA is denatured into two single-stranded DNA. At stage 2 is annealing of DNA primers to the single-stranded DNA templates. The reaction which is done earlier, which was done at 95 degrees, is cooled to 50 to 60 degrees centigrade for a minute. That allows annealing of the primers present in the reaction mix to the complementary sequences in the DNA. The primers attach to a starting point on single-stranded DNA template. Primers are single strands of DNA that are about 20-30 bases in length, as I already explained before. The cartoon at the bottom describes the small piece of primers, how they anneal to the template DNA, single-strand template DNA, through complementation. At a forward primer, anneals at the near close to the 5' and the reverse primer also anneals at close to 5' at the reverse strand. The third stage of PCR is extension. Once the primers anneal, these primers need to be extended. During this final step, the temperature is adjusted to allow Taq polymerase to synthesize complementary strand by using 3' OH group of the primers. The duration is usually 2 minutes at 72 degrees centigrade. Taq polymerase is a heat stable and it is isolated from the bacterium from thermal springs, which is known as, this bacterium name is Thermus aquaticus. This is an enzyme which can withstand high temperatures up to 95 degrees centigrade or more, even 98. And it can pass through several cycles of low and high temperatures still without getting denatured. These three stages of PCR are repeated 20 to 30 times, which results in production of large quantities of DNA product. Second step in gene cloning is insertion of isolated DNA fragment into vector. The isolated fragment can from restricted DNA or from DNA libraries and so on. How this gene of interest can be inserted into a vector. To start this, we should know first what is a vector. Vector is a vehicle for gene cloning. A vector is a DNA molecule that has the ability to replicate autonomously in an appropriate host cell and into which a DNA fragment to be cloned is integrated. What are the properties of a good vector? It must be able to replicate within the host cell so that numerous copies of the recombinant DNA molecule can be produced and passed to the daughter cells. A cloning vehicle should be relatively small, usually less than 10 kb in size, as large molecules tend to break during purification steps. The vector should have suitable selection marker gene for easy detection of transformed cells. Two kinds of DNA molecules that satisfy these criteria are plasmids and bacteriophage chromosomes. Plasmids are circular molecules of DNA that lead an independent existence in the bacterial cell. Plasmids almost always carry one or more genes, and these genes are responsible for a useful characteristics displayed by the host bacterium. For example, the ability to survive in normally toxic concentrations of antibiotics such as ampicillin is often due to the presence of the bacterium of a plasmid carrying antibiotic-resistant gene. In the lab, antibiotic resistance is often used as a selectable marker to ensure that bacteria in a culture contained particular plasmid. Most plasmids possess at least one DNA sequence that can act as an original replication. That's called origin of replication. So they are able to multiply within the cell independently. Plasmids are independent genetic elements found in bacterial cells. An example is shown here. This plasmid is one of the types of vectors. This shown here on the right side, RP4. This plasmid carries one or to several genes. In this case, 3 genes are shown. There's a particular plasmid, synthetic plasmid. It contains kanamycin gene for kanamycin resistance, gene for tetracycline resistance, and another gene for ampicillin resistance. A bacterial cell transforms this particular vector, expresses 3 different proteins to neutralize 3 different types of antibiotics. And when this type of vector, a vector containing antibiotic resistant genes, are transformed into bacterial cell, and the bacterial cell acquires the ability to grow on a medium containing all 3 different antibiotics. In this case, a medium containing tetracycline, kanamycin, ampicillin can be used to select the transformants containing this particular vector. In other words, the use of antibiotic resistance as a selectable marker for a plasmid, as I just mentioned, RP4 carries genomes for resistance to ampicillin, tetracycline, and kanamycin. Only those E. coli cells that contain RP4 are able to survive and grow in the medium that contains toxic amounts of one or more of these antibiotics. You can see this slide represents it's included to show that these plasmids come in various sizes. For example, on the first column, the names of plasmids are given here. On the left side, PUC8 is a small plasmid. It's about 2.1 kb in size. Molecular mass is 1.8 megadaltons. It can be replicated in E. coli. In fact, most of these can be replicated in E. coli, but these plasmids, which were originally isolated from various organisms, are listed on the last column on the right side. You can see here, there's another plasmid, PTI-ACH5. This is from Agrobacterium tumefaciens. It's a considerable, very large plasmid containing about 213 kb genome, and molecular mass is 142 megadaltons. Other plasmids are listed. There are many more, hundreds of plasmids with different characters and different sizes. An important part in this insertion of the gene is ligation. How you can join vector molecule with the insert DNA molecules. Ligation is nothing but joining of DNA molecules together. The enzyme that carries out this ligation reaction is called DNA ligase. DNA ligase used in genetic engineering is usually purified from E. coli bacteria that have been infected with the T4 virus. Ligation is the final step in construction of a recombinant DNA molecule. Here, the picture at the bottom shows there's a vector and a gene fragment. Both are mixed together in presence of DNA and ATP and an appropriate buffer containing ATP. They are incubated at a low temperature, 4 to 14 degrees, whatever, depending on the type of enzyme you are using, efficiency of enzyme. And the ligase reaction, when it's performed, the DNA insert is joined with the vector molecule, forming a recombinant DNA molecule. 2 different types of joining reactions are catalyzed by DNA ligase. One is ligation of blunt-ended molecules. As shown in the picture here, A, ligating blunt ends. 2 blunt-ended DNA molecules can be ligated. A phosphate bond can be formed in both strands. And there's another type of ligation, is the ligation of sticky-ended molecules, as we already talked. Like for example, ECHO R1 or BAMH1 creates, when it cleaves, restriction enzyme cleaves the DNA, it forms a sticky ends. Sticky ends are easy to efficiently ligate because complementary sticky ends come together. the vector as well as the insert ones. They form a sort of a hydrogen bonds between them, complementary strands, and the discontinuities are filled in by ligase by breaching them with the phosphate bond. Sticky ends of a DNA molecule can increase the efficiency of ligation. Ligation of sticky ends is much more efficient because sticky ends can base pair with one another by hydrogen bonding, forming a stable structure for an enzyme to work on. Therefore, sticky ends are desirable on the DNA molecule to be ligated together in a gene cloning experiment. Sticky ends can be put into a blunt-ended molecule by using linkers or adapters. Linkers and adapters are used to make the cloning easier. For example, if you have only blunt-ended DNA, you can use the linkers and join them together and cleave them and clone the appropriately digested vector. Linkers are short pieces of blunt-ended double-stranded DNA that contain a restriction nucleus, a restriction site. A typical linker is shown here. It's a double-stranded DNA. It's a blunt-ended on both sides. It is embedded with the BAMH1 site here. And this restriction site can be cleaved by BAMH1 to create a sticky ends. The attachment of linkers to blunt-ended molecule is done by ligase reaction, DNA ligase. Linkers are joined together and form a concatenamer along with the blunt-ended DNA molecule to which they are ligated and is digested with, in this case, BAMH1 gene, so that a DNA molecule with a blunt-ended one can be turned into a one which has got a sticky end because the linkers which are attached are digested, leaving one linker each on each side. Second type of molecules is the adapters. These are also short synthetic oligonucleotides, but unlike linkers, an adapter is synthesized such that it already has one sticky end. A typical adapter is shown in this picture here, and this particular picture has got a BAMH1 sticky end, a DNA molecule which has already been digested by BAMH1. There's an overhang of GATC here, you can see, and the upper strand. The problem with using adapters is that the two adapters could ligate to one another to produce a molecule similar to a linker. Both sticky ends to complement and can be ligated. So that after ligation of adapters, a blunt-ended molecule is still a blunt-ended, and the restriction step still need to be performed. As shown in the picture at the bottom, on the right side is two adapters coming together, complementary sequence. If you concentrate on GATC sequence, and they form complementation, and the discontinuities are sealed by phosphate bond through the action of ligase. And if you, on the right side, you can see a DNA molecule mixed with linkers and ligated. And the linkers joined with the linkers can be ligated, can form a concatenamer, and some such things can also join with the blunt-ended DNA molecule to have a sort of a larger molecule with adapters fused to it, which again, because of self-ligation of linkers, form blunt-end. These need to be again digested with appropriate restriction enzyme, in this case BAMH1, to create sticky ends for cloning purposes. The third step of cloning is transformation. Once you developed a recombinant DNA molecules, inserted a gene of interest into a vector, it needs to be transformed into a host cell. That is, the transformation nothing but introduction of DNA into host cell. Most species of bacteria, including E. coli, take up only limited amounts of DNA under normal circumstances. In order to transform this species efficiently, the bacteria have to undergo some form of physical and or chemical treatment that enhances their ability to take up DNA. Cells that have undergone this treatment are said to be competent. Treatment with the calcium chloride makes them permeable to DNA, and for transformation, DNA is mixed with these competent bacterial cells, and since the calcium chloride makes the cell's competent, cell wall is made permeable or brittle, and also calcium chloride, calcium molecules encourage the attachment of DNA plasmid molecules on the cell wall surface, and this mixture is heated to 42 degrees for about a minute or two, so that the plasmid is, and put it in ice, transferred to ice, that's a 4 degrees. The plasmid which is attached to surface is sucked into inside the cell, and that cell is known as transformed cell. The last step of cloning is, once the cells are transformed, bacterial cells, how to select which cells are transformed, which are not transformed. What happens is, in addition to desired recombinant molecule, the ligation mixture may contain unligated vector molecules, which are clearly shown on the right side of this slide, and unligated DNA fragments, self-ligated vector molecules, recombinant DNA molecules that carry the wrong inserted DNA fragment, or recombinant molecules having a desired recombinant insertion, a DNA insertion. So when you take this mixture of different recombinant molecules, DNA molecules, and transform E. coli cells, the complete vectors, plasmids, along either with or without insert or transformed inside the bacterial cell, as we discussed in earlier slide, transformation. And all these cells, since they have the vector, they can be grown on a selection medium containing an appropriate antibiotic, for which the antibiotic resistance gene is located on the vector itself. So all self-ligated vector, the clone with the desired gene, a clone of a wrong insert, all develop into colonies. How to distinguish transformed cells from non-transformed ones? The plasmid taken by the host cell during transformation carries a selectable marker. A selectable marker is a gene that gives transformed cell a new characteristic that is not possessed by a non-transformed cell. For example, ampicillin-resistant gene of PBR322. After transformation with PBR322, only those E. coli cells that have taken up the plasmid are ampicillin-resistant and tetracycline-resistant and form colonies on agar plate contain ampicillin as well as tetracycline. The non-transformed cell will be ampicillin and tetracycline sensitive and do not produce colonies on the selective medium. Therefore, transformed and non-transformed cells are easily distinguished. As I explained earlier, selecting cells that contain a particular plasmid, in this case, we have taken an example of PBR322 plasmids, by plating onto agar medium containing ampicillin and or tetracycline, two different antibiotics. Normal cells with no plasmids cannot grow on the medium containing either ampicillin or tetracycline or both. Whereas the cells which received the recombinant molecules or empty vector of PBR322, when they are transformed, those cells can survive on the medium containing ampicillin or tetracycline. That's how one can identify the clones. Identification of recombinants. Insertional inactivation is the most common way to identify the recombinant molecule, correct recombinant molecule. First one is insertion inactivation. Works on the principle that the characteristic function coded by the gene that has been inactivated is no longer displayed by the host bacterial cells. In other words, a gene, once it is interrupted, the gene gets inactivated and its phenotype is not displayed anymore in the bacterial cells which receive this plasmid. For example, PBR322 has a unique restriction site that can be used to linearize the vector. PBR322 has one restriction site for BAMH1 within the gene that code for tetracycline resistance. A recombinant PBR322 clone, which carries one extra fragment of DNA at BAM-H1 site, will no longer be able to confer tetracycline resistance. The recombinant cells will still confer resistance to ampicillin because that ampicillin gene is not interrupted, and it becomes sensitive to tetracycline. It is shown here, an example, fashion of a cartoon. The normal vector molecule is shown, and the target gene, when it's interrupted at a particular site, a BAM-H1 site, when the target insert is being incorporated into the vector, tetracycline gene is disrupted and it gets inactivated. The normal non-recombinant vector molecule carries a gene whose product confers selectable or identifiable characteristic on host cell. This gene is disrupted when new DNA is inserted into vector. As a result, the recombinant host does not display the relevant characteristic, as we have already discussed about. An example of PBR322 we have discussed here, and it's pictorially represented at the bottom of the slide on the right side. Another example of insertion and inactivation is identification. For identification recombinants is blue-white colony selection. Clone experiment with the PUC-based vectors, in this case PUC8, involves selection of transformed clones on ampicillin plate followed by screening for beta-galactosidase to identify recombinants. PUC8 carries ampicillin-resistant gene and a gene called LAGC that codes for the enzyme beta-galactosidase. Expression of the LAC operon is induced by lactone in natural conditions and also by a lactose analog IPTG, that is isopropyl beta D1 thiogalactopyranoside. When expressed, the beta-galactosidase enzyme can break down, a complete enzyme can break down a dye-linked substrate called XGAL. That is, chemical name is 5-bromo-4-chloro-3-indolyl beta-D-galactopyranoside. This particular compound is broken down into galactose and an insoluble blue pigment called 4-chloro-3-brome indigo. Colonies with an insert containing vector have a non-functional beta-galactosidase because it is interrupted by insertion of the DNA molecule. The gene is inactivated, so non-functional beta-galactosidase is produced, and these colonies which receive this vector remain whitish cream in color. On the other hand, intact beta-galactosidase produce a pigment from XGAL included in the transplantation, turning the bacterial colony into blue. When the beta-galactosidase gene is not interrupted by insertion of a gene of interest, which is called exogenous gene, then this gene, since it's not cloned into this galactosidase, this will remain a fully functional beta-galactosidase, hence is able to cleave XGAL into two products. One of the products is the blue pigment. What is the rationale behind using blue-white screening? It is very clearly demonstrated here. Beta-galactosidase is produced by a particular gene called LAGC. It is artificially cloned into a plasmid. Part of this gene artificially cloned into a plasmid, and it is part of is left in the bacterial genome. And that means this enzyme, since it is coded separately into two different fragments, both the fragments should combine together, form an active protein. So the LAGC gene, which is cloned in the vector, that's the gene, when it produces partial beta-galactosidase, it complements the wild-type partial beta-galactosidase part and forms a full complement of beta-galactosidase molecule, which have got the ability to cleave XGAL to produce blue. If there's insertion LAGC, that LAGC gene cannot produce a functional beta-galactosidase. Then the bacterial cells which receive this plasmid, they show the white color, whereas the transformants with intact beta-galactosidase show blue color in colors. In today's lecture, I tried to explain basic aspects of gene cloning, transformation, and selection of recombinant organisms. In the last 40 years, molecular cloning has progressed from laboriously isolating and bringing together two pieces of DNA, followed by assiduously screening of potentially important clones, to perfectly assembling up to 9 to 10 DNA fragments with remarkable efficiency in just few hours, or designing DNA molecules in silico and synthesizing them in vitro. Together, all these technologies give molecular biologists an astonishingly powerful toolbox for exploring, manipulating DNA that will further broaden the horizons of science. Thank you.